this crash course, I divided the discussions into two parts. So for this video, we will tackle first your lipid metabolism. And for our part two, of course, that will be your amino acid metabolism discussion. Class, remember here in your lipid metabolism, we will first discuss the overview, including your lipid digestion. Then next, your lipid catabolism, or mainly your fatty acid catabolism. And of course, if we have your catabolic pathway, our third topic here is your biosynthesis. And of course, for our special topic, we will discuss your ketogenesis, our alternative source of energy. Okay? And of course, class, I also added a discussion on cholesterol biosynthesis. Oh, sir, does that topic appear in the board exam? Yes po. But don't worry, class, I summarized all relevant information on your cholesterol biosynthesis. Already na? So of course, class, if you're ready, I'm going to proceed with our discussion on lipid metabolism. So class, before we begin discussing our topics for today, so let's just have a quick recap of what we have discussed so far. Remember class, hopefully hindi kayo nagkaka-amnesia. So again, for our first two videos, we tackled your biomolecules, carbs, lipids, proteins, including your enzymes, and nucleic acids. Remember, we highlighted or we focused on their structure, their function, and their key properties. Alright, sa ating third and fourth video, we discussed your intro or principles to metabolism or your meta pathway, as well as the biological membrane and membrane transport. Or recall, diba, I have emphasized there that your glycolytic pathway is a part of your cellular respiration and as well as your carbs metabolism. Recall also that in our fifth and sixth video, we discussed the whole pathways under your carbs metabolism. And for this video, we will start with our lipid metabolism, particularly your catabolism, your biosynthetic pathway, and of course, your ketogenesis, our alternative energy source during low carbs or low blood glucose concentration. On a separate video class, remember, we will tackle naman your amino acid metabolism. Don't worry, I will highlight and emphasize the relevant info that you really need or you are really required to memorize or familiarize the concept. Okay? So I hope that's clear. Again, these are the five or the pathways rather that we discussed on your carbs metabolism. So the glycolytic pathway, we separately discussed that on our cellular respiration process. So our first pathway here is your PPP, wherein we produce your pentoses for your nucleic acid biosynthesis. And of course, we have also seen your another product, erythrose 4 phosphate. Also, can you recall what is the use of your T4P nga? Again, for the synthesis of your amino acids. And aside from that class, remember, we produced here your reducing equivalents or reductants, your NADPH, that will be used for majority ng biosynthetic pathways. So class later, we will emphasize the difference, your NADH and your NADPH in terms of the pathways they participate to. For the meta pathway, si PPP po ay amphibolic. So for the catabolic process, of course, it produces your reducing equivalents. For our anabolic, of course, building up molecules via the synthesis of your pentoses, and of course, additionally class, your glycolytic intermediates that will be fed if our body or our cells need energy in the form of ATP. Ayan, for the location class, remember, your PPP is located in the cytosol because of the availability of your initial substrate, your G6P coming from the first reaction ng ating glycolytic pathway. And of course, here, remember, I emphasized our key or the committed enzyme is glucose 6P dehydrogenase. And here are the list of the products. So here class, remember, we produce a byproduct, carbon dioxide. Okay, so for the next two pathways, we know that these are essentially the reverse of each other, diba? First one, glycogenolysis, from the name itself, lysis, meaning we will degrade your glycogen to mobilize your glucose source. Of course, class, from the term itself, we know that it is catabolic because in reality, this glucose kasi will be fed in your glycolytic pathway to produce energy in your TCA cycle. Class, for both of these processes, they occur in your cytosol. Similar reasons lang yan again, due to substrate availability. Okay? Ang ating key enzymes for these two pathways, you have your glycogen phosphorylase for glycogenolysis. On the other hand, synthase for glycogenesis. Class C, they are just really the reverse of each other. And for our products, glucose for your glycogenolysis and glycogenesis, we have your birth of glycogen, diba? Your glycogen polysaccharide. Class, for our fourth pathway, we have your GNG. So, diba, we have studied, we have discussed, this is actually essentially the reverse of your glycolytic pathway. In a way that, of course, there are reactions that are similar to both pathways. Of course, we need to remember the bypass reactions. So, in our glycolytic pathway, we have three reactions bypassed in your GNG via the four different reactions. Class, remember, our precursor here is your non-carbohydrate precursors, diba? So we have your lactate, we have your glycerol, and we have your alpha ketoglutarate. And remember, all of them will be eventually converted first to your pyruvate, our starting molecule for GNG or gluconeogenesis. Class, again, remember, here, unique po ang GNG because among the four pathways here, it is only the pathway that is located in your both cytosol and mitochondria. Okay? So for our key enzyme, we have your fructose 1,6-bisphosphatase. O sir, bakit siya po yung enzyme na regulated? Remember, this enzyme is actually the bypass enzyme of your PFK1 ng ating glycolytic pathway. And of course, we know the committed enzyme in our glycolytic pathway is that PFK1 enzyme. That's why essentially this is also your committed step or committed enzyme for GNG. And clearly, class, our product here is glucose. And lastly, for our photosynthesis, remember this is very unique for our photoautotrophs, or essentially for plants or plant organisms, particularly in their chloroplast. Class, remember, your photosynthetic pathway has two phases, the light-dependent and your light-independent, or we also call it your dark reactions, your C3 photosynthesis, or your Calvin cycle or Calvin-Benson cycle. Okay? So here class, they are both anabolic because eventually we will produce glucose as our general plant product for photosynthesis. For the location, we have your thylakoid membrane for photosynthesis. For the light-dependent class. And for your Calvin cycle or dark reactions, we have your stroma. Remember, diba? I will give you a mnemonic here during our practice test discussion. Class, for the key enzyme, just remember the enzymes that catalyze the production of your energy products for the light-dependent reaction. Again, class, ATP synthase, both your cyclic and your non-cyclic photophosphorylation will use ATP synthase. Diba, si cyclic, it only produces ATP exclusively. Si non-cyclic, of course, aside from ATP, it can also produce NADPH and your oxygen molecule that will be released into the air. Alright, class, the NADP plus reductase, it is also regulated because again, this is the enzyme that produces your NADPH reducing equivalent. And finally, for our Calvin cycle, remember the unique properties ni ribisco is its carboxylase and oxygenase functions or activity. And here, We converted your starting reactants, particularly si carbon dioxide and water, as well with an input, of light energy with our sunlight as our source of light energy to form glucose or G3P class. So of course, these are both carbohydrate or monosaccharides. Okay, so I hope it's already clear in your side class our discussion here on carbs metabolism. Okay, and we proceed with our focus topic for this video. So again, we will discuss here your lipid metabolism. Okay, so class, again, remember in lipid metabolism, this is a pathway that encompasses both your catabolic and anabolic processes. So for catabolic, we have, yes, breaking down of fats for energy source. And of course, for biosynthetic pathway, remember, we have two different pathways. For making your fatty acids, we have your biosynthesis. And for our ketone bodies, we have your ketogenesis. So class, always remember, ang synthesis ng fatty acids, it occurs for your storage, okay, for energy storage. And on the other hand, we synthesize your ketone bodies through ketogenesis when we have low blood glucose concentration or during fasting. Let's check our concept map here that summarizes the whole process of lipid metabolism. Let's check. First class, this is your either dietary tags or endogenous tags or your triacylglycerols synthesized by our body. So of course, class, ang ating dietary tags will first be digested, okay? So of course, this is a process which we will discuss later on. And of course, ang reverse niyan, that's your biosynthesis ng triacylglycerols coming from your fatty acids. Di ba remember sa carbs metabolism, ang main character po natin si glucose. Ngayon naman, of course, our focus here is your fatty acids. And class, remember, aside from our energy role ng lipids, particularly ng fatty acids, we also have its structural role, okay? So of course, the fatty acids can also be used in order to form spontaneously our membrane lipids, okay? So it can also aid your structure and as well as for transport ng ating biomolecules. So remember, si phospholipids, they are the principal component ng bilayer membrane natin. So it will just be integrated or be reacted with our phosphate groups to create your phospholipid bilayer membrane. Let's check naman the full process for our fatty acid. Or coming from your fatty acid, of course, we have your catabolism. So class, your catabolism is an umbrella term. Your beta oxidation is just a part of your catabolic process of your fatty acid. So later on, we will rationalize why do we call this process beta oxidation of fatty acid. Check, may teaser na tayo. Ang product natin under your beta oxidation is your acetyl-CoA molecule. From our cellular respiration discussion, I emphasize this molecule as your central energy molecule because it will be eventually fed a citric acid cycle. So if we have large amounts of energy, of course, it will be diverted to other pathways, starting with your biosynthesis. So we will discuss your biosynthetic pathway later on. Of course, after our catabolic pathway. Class, check. Our fatty acids can also serve as energy storage. So actually, it is stored in our fat tissue, or we also call it your adipose tissue. And aside from that, we also have your cholesterol biosynthesis. Sir, ano nga po ulit ang biological role or relevance ng cholesterol? Remember class, it's a part of your membrane lipids, di ba? So it gives your bilayer its fluidity in order to have that selective permeability and in order for it to have your lateral or rotational movements. Clear? So last one here is your ketogenesis. So of course, your ketogenesis will produce ketone bodies. And class, later on, we will see why our cells have two pathways in order to store your energy. And of course, our reason here is rooted from the fact of fatty acids being hydrophobic. Okay? So again, your ketone bodies are efficiently packed in order to store our energy, of course, efficiently, diba? So of course, class, again, that's the concept map for lipid metabolism. So let's tabulate those pathways that we will discuss later on. Again, we will start with your catabolic pathway. Here, our main role is to produce energy via the formation of your acetyl-CoA. Class, again, we will break down your fatty acids to form your acetyl-CoA. Remember, this is a complex process. Class, it's not a straightforward reaction. I think you can remember it in your discussions sa undergrad biochem. And again, when our cells need energy, your acetyl-CoA will be fed in your citric acid cycle. And class, essentially, of course, your reverse ng catabolic pathway or your FA catabolism is your FA biosynthesis, okay? So here, from our acetyl-CoA, we will store it as fats for energy source for future needs, okay? So in particular, your fatty acids will actually be condensed pa nga sa ating glycerol backbone to form your triacylglycerol that will be stored in our adipose tissue as well, okay? For our third pathway here, we have your ketogenesis. So class, again, these two pathways, biosynthesis and ketogenesis, they serve as both energy source during cells needs, okay? So again, si ketogenesis, this is an alternate energy source during fasting or when we have low blood glucose concentration. So of course, coming from acetyl-CoA, we will generate your ketone bodies. Class, when we say peripheral use, your cells will only use it aside from the location of your ketogenic pathway. Okay, that's very important for efficient energy mobilization. Clear? Alright, so for the last one, so for cholesterol biosynthesis, I know that this is relatively a hard pathway. That's why in our discussion class, don't worry. So we will just summarize the relevant information. Class, wag matakot na. Again, your cholesterol biosynthesis, very straightforward lang ang questions coming from these pathways. And we will highlight it later on during our discussion. Again, main role for sterol production, for instance, for our membranes and hormonal production. And again, for our cholesterol biosynthesis, our main precursor molecule is your acetyl-CoA to your production ng cholesterol and its derivatives such as your hormones or particularly your sex hormones. Alright, so again class, as you can see, for our biosynthetic pathways, FA biosynthesis, ketogenesis, and cholesterol biosynthesis, we mainly use acetyl-CoA as our main precursor molecule. But class, remember, these three processes are still regulated. So class, let's start our discussion on your catabolic pathways in order to degrade your dietary tags to get your individual fatty acid molecule. So again, class, ang fatty acids, ang main role niya inside our cells is for energy use, okay? But of course, always remember, we still prioritize your carbohydrate as our main energy source. Let's check our overview for this topic. Ayan. So as you can see here, our specific process here involves the sequential removal of your 2 carbon units. Remember, diba? Our product here in your catabolic pathway is acetyl-CoA. And recall the structure of your acetyl-CoA, it is a 2 carbon compound. And of course, it will be coming from your fatty acids as our energy source. Okay? clear? So again, class, from the name itself, it's very obvious that this is a catabolic process. For our products later on, you will see we will yield large amounts of your energy in the form of acetyl-CoA, NADH as our electron carrier, and FADH2. And lastly, ayan, for our location, remember, it is located in the innermost part of your mitochondria, particularly its matrix, because again, class, the enzymes that catalyze all energy-producing pathways are always located, or majority of them are located in your matrix. Class, remember, our energy-yielding pathway as well are or is concentrated in your liver and muscle. Okay? Because of that, ayan, we will now discuss the specifics of our fatty acid metabolism. Let's check the specific processes under our metabolic process. So our discussion will start with lipid digestion. So class, again, later we will see the sites of our lipid digestion. And then of course, afterwards, we will get kasi your fatty acids here after your lipid digestion. And of course, we need to mobilize it for storage or energy use. If it's for energy use, of course, we need first to activate your fatty acid for transport to your mitochondrial matrix. And lastly, again, our main site of your beta oxidation, ayan, is your mitochondrial matrix. So let's start, class. So I hope you are very eager for this topic. So during my review, actually, this is somehow one of my favorite topics because it's relatively easier to digest, very straightforward on reactions, and of course, it's fun to discuss. Okay, so let's start with our lipid digestion. Class, again, ang main source ng ating lipids is through your dietary tags or endogenous tags stored in your adipocytes or adipose tissue, a region of your adipocytes. Okay, so class, here, see our main site of lipid digestion actually begins in your small intestine and it's only minimal in mouth and stomach. So later on, we will see time out. That's just your mechanical digestion. And of course, sa stomach, very rare ang digestion or beginning ng digestion in our stomach. Or remember our discussion on enzymes. The main enzymes that can degrade your lipids, particularly your dietary tags, are your pancreatic lipase and bile salts. But class, please remember, we have different types of lipase and they have different functions in terms of the structure or the biomolecule that they can process or degrade. And lastly, here our products after lipid digestion or after your hydrolysis is 2 free fatty acids plus monoacyl glycerol, or we call it your MAG. O class, again, ang daily intake, if I'm not mistaken, ang recommended daily intake for your total fats are around 60 to 150 grams a day. Okay, so of course, if we consume excessive amounts, it will be just stored in our adipocytes or our adipose tissues. Okay, so class, that's why we also need to regulate our lipid intake or our fat intake. Okay, so class, going back, here is our overview of our hydrolysis process occurring in order to degrade your tags. I recall the structure of your triglycerides or tags. So as you can see here, we have your glycerol backbone plus your 3 different fatty acids. They can be unsaturated, they can be saturated. So class, not necessarily similar ang structure nila to each other, ha. So again, upon hydrolysis, we will yield 1 molecule ng monoacyl glycerol. So as you can see, we retained your MAG here plus 2 free fatty acid molecules. So of course here, we cannot cleave your fatty acid at position 2 because of steric hindrance and enzyme orientation. That's why here, class, ang main enzyme natin is si pancreatic lipase only. So very selective ang hydrolysis mechanism ni pancreatic lipase. So it can only hydrolyze your ester bonds at the C1 and C3 positions. Okay, so I hope that's clear. Again, selective hydrolysis ang mechanism ni pancreatic lipase. So let's check the sites of our digestion. First one, ayan, mechanical digestion in order to increase the surface area of our food molecule. In the stomach, again, as I have said, very rare, okay? Because of the acidic environment in our stomach, your fat will form droplets. And of course, this will be mixed with our food enzyme. That's why it is called your chyme. And lastly, in the small intestine, again, is our initial or main site ng digestion. Oh, sir, paano po? Of course, we have a hormone here. We have your cholecystokinin or CCK in abbreviation. So class, again, this is a hormone that will trigger the release of our bile salts and your lipases. So si pancreatic lipase and bile will help in degrading our triacyl glycerol, okay? So si bile salts kasi, class, such as your sodium taurocholate and glycocolate has an emulsifying property because of its hydrophobic and hydrophilic portions. Okay, so again, they act as an emulsifier, making insoluble substances soluble in water. Okay, so again, we will degrade your triacyl glycerol first through the help of your lipase, and si bile, it will protect its structural integrity through forming your micelles. Okay, so again, class, it will act as an emulsifier. All right, so class, let's delineate or let's compare the different types of your lipases. So again, we have 3 different types ng lipases, and they are very unique or very specific in the reactions they catalyze. So let's see. Si pancreatic lipase po, again, the only mechanism of this type of lipase is to break down your dietary tags into free fatty acids and monoacyl glycerols. Okay? Si hormone-sensitive lipase naman, ayan. Ito naman, class, ang lipase natin responsible in degrading your tags stored in adipose na. Okay? So again, ang boundary, di ba parang jeep? Ang boundary ni pancreatic lipase is the degradation ng dietary tags sa intestine. Okay? Because again, it is mainly secreted by our CKK or your cholecystokinin or your CCK, sorry, your cholecystokinin molecule. Si hormone-sensitive lipase, ayan, once we stored your tags coming from the intestine or the one we synthesized in the body, your hormone-sensitive lipase will break it down if our cells need energy. Okay? And si MAG lipase, finally, again, class, this is very specific sa cleavage or your hydrolysis reaction ng MAGs to form your glycerol and fatty acids. Okay? Again, that's a very specific reaction. And of course, it can occur in your small intestine and of course in your adipose tissue or adipocytes. Class, here is the specific reactions occurring in our lipase mechanism. So again, starting with our tags, your pancreatic lipase will induce hydrolysis to create your 2,3 dags. O sir, ano po yung 2,3 na prefix? Again, that's our site ng acyl groups. Okay? So of course, class, ang unang i-cleave is the C1 position. Readily available kasi siya. And then here, we liberate your free fatty acid. Thus, this is actually a stepwise reaction. After we yield your 2,3 dags, we will further hydrolyze it in order to get your final product here for your pancreatic lipase, 2 monoacyl glycerol plus another molecule ng free fatty acid. That's why here we were able to liberate 2 free fatty acid. And of course, consequently, si monoacyl glycerol will be hydrolyzed by your MAG lipase in order to liberate your full glycerol molecule plus another molecule ng free fatty acid. O class, remember, si glycerol, it has also a different fate coming from your lipid digestion, okay? So your glycerol actually can serve as a precursor or a source of glycolytic intermediate. Talaga ba, sir? Yes, check your manual, okay? So of course, we will also discuss it here in our lipid digestion discussion. Let's read. Your free fatty acids actually coming from your lipase digestion are reassembled and packed into lipoproteins for mobilization na class, okay? So that's our second step. So we will discuss the details of this reassembly and your repackaging, okay? So si glycerol class, again, please remember, it can enter either your glycolytic or your gluconeogenic pathway. So again, we can switch on and off these two pathways depending on our cell's need. So class, remember the term lipolysis. So this is just the metabolic breakdown of your tags into free fatty acids and glycerol. So simply, that's what we have discussed from our previous slides, okay? So class, here in the slide, let's discuss our glycerol pathway. So here again, we will convert your glycerol, the backbone of your tags, to your G3P for glycolysis or GNG or gluconeogenesis, okay? So let's see the specific reactions. So check our main enzymes here. First, we have your kinase molecule or kinase A enzyme, your glycerol kinase, and your glycerol 3-phosphate dehydrogenase will be for the redox reaction naman. For our location class, our glycerol pathway is located in your cytosol. And here, we will use initially your ATP in the first reaction and NAD plus as our electron carrier in our subsequent or second reaction. Let's discuss briefly the reaction mechanism. First one, the glycerol coming from your tags, I'll check the structure again, will be phosphorylated through your glycerol kinase reaction. So class, this is the main committed enzyme for your glycerol pathway. Sir, and I ask a question, do we need to memorize the structure? No na po, okay? So here, just remember our essential information given here in our table. Going back, here, we were able to form your glycerol 3-phosphate molecule which will be subjected to a round of a redox reaction, okay? So here, we will yield an NADH, one molecule of NADH per glycerol molecule. And here class, ayan, sir, familiar na yan, glycolytic intermediate na po siya. Yes, that is your DHAP. That's why si triose phosphate isomerase is the fourth reaction in our glycolytic intermediate. And of course, it's also in your gluconeogenic pathway. And here, our product is your glyceraldehyde 3-phosphate. Already na yan for energy generation in your glycolysis and TCA cycle and for production naman ng glucose in your gluconeogenic pathway. Class, here, again, si glycerol, it has a different fate compared to your 3 free fatty acids. Okay? So now we are done with our lipid digestion. Class, breathe muna. So again, at this point, very saturating na ang discussion natin. You are super saturated students na. Class, again, as we have emphasized earlier, after we digested your lipids, we will get your free fatty acids and glycerol. However, in order to efficiently mobilize it, sometimes we repack it again into your triacyl glycerol after we emulsify it via your bile salts mechanism. Okay? So here, after repacking, we will form your lipoproteins in order to deliver our tags to tissues. Let's see the main mechanism here. From your small intestine, again, we will form your fatty acids plus MAGs. So class, there are times, particularly when we need really large amounts of glycolytic and gluconeogenic intermediates, si glycerol will be used by the body. However, if we are going to store it, ayan, it will pass through your membrane to reach your mucosal cell where it will be converted back to your tags, plus integrated or condensed with other lipids and proteins in order to protect its structural integrity via the formation of your lipoprotein. Or from the name itself, again, class, your lipoprotein consists of lipids and protein components. Sir, bakit po need i-convert si tags rather than directly transporting it to your membrane? Class, your intact tag or your dietary tags cannot pass through your membrane going to your mucosal cells. Okay? That's why we need first to hydrolyze it via the production of fatty acids and MAGs for efficient absorption ng intestinal cells. Okay? So class, again, for lipoproteins, we have different types. And later on, we will discuss those depending on their roles in our lipid mobilization. Class, here is an electron microscope picture of our lipoprotein granules. As you can see, very concentrated in our adipocytes, see lipoprotein. Okay? So class, again, your lipoproteins, including your chylomicrons, so this is a type of lipoprotein, are lipid plus protein complexes that transport fats in the blood for delivery in different parts of our body. Okay, class, again, let's discuss. For our lipoproteins, always it constitutes your triacyl glycerol, your cholesterol, and phospholipids. So class, actually, these three main lipids are the one being delivered to our different parts of the body or to your liver. Okay? Also, how about your lipoproteins or your apolipoproteins? So class, this actually serves as our main cell receptor. So it's on the surface of your lipoproteins and it binds to your specific sites for delivery and acceptance or weathering for modification ng lipoprotein. So again, this is very important for cell signaling. Okay? So here in our slide, we have your first two main types ng lipoproteins. Starting with your VLDL or familiar, diba? We know that this is a very unhealthy lipoprotein. Okay, so let's discuss. Its main function is to deliver your triacyl glycerol stored to your adipose tissue to your other tissues. Okay? So sir, bakit siya tinawag na low density? As you can see here in the slide, very high daw ang triacyl glycerol. Class, remember, the name here is with respect to your protein component. So low density po kasi ang protein component ng VLDL. Okay? Thus, if it has a very low density ng protein component, it also has a very high tags content naman. That's why it is also called your highly atherogenic lipoprotein. When we say atherogenic, class, meaning it can contribute to build up ng fatty plaques, particularly sa arteries natin. Okay? So of course, we want to avoid that build up ng fatty plaques. That's why as much as possible, we do not want VLDL to accumulate in our biological system. So let's proceed with our intermediate density lipoprotein. From the name itself, intermediate lang yung protein component in terms of its concentration inside your lipoproteins. Okay? So of course, class, after delivery ng tags, mababawasan yung lipid component. That's why it is your remnant ng VLDL. So your intermediate density lipoprotein is the product upon delivery ng tags to your tissues. Okay? So here, it is your VLDL remnant and LDL precursor. Meaning, upon delivery ng lipid component here, we will form your LDL or your low density lipoprotein. Again, it has moderate tags component or your content and it's potentially atherogenic because of your triglyceride and cholesterol content. Clear ba, class? For our last two lipoproteins, we have your LDL and HDL. Class, ang very unique kay low-density lipoprotein is that it has a very high amount ng cholesterol content. That's why this is actually our major atherogenic lipoprotein. Class, a very unhealthy CLDL. Again, this is coming from your IDL. So this is just really a process of delivery of your lipids. So from our VLDL, kapag na-deliver na ang tags, we will form your intermediate density lipoprotein. Once we deliver the remaining tags, ayan, we will only be remaining with our high cholesterol content in our LDL. So again, it delivers mainly your cholesterol to cells for your phospholipid bilayer membrane. Again, your cholesterol is a component of our lipid bilayer. And of course, ayan, ang very healthy lipoprotein natin, your high density lipoprotein, ang main or ang unique property nito is its low cholesterol and high protein content. That's why it is called your high density lipoprotein because it lowers your cardiovascular risk through reversing the cholesterol transport of LDL. So of course, class, if it's reversed, it delivers cholesterol from your cells to your liver naman. So of course, yung mga excess cholesterol delivered by your low density lipoprotein will be delivered back to your liver by HDL. Kasi class, yung excess cholesterol can contribute to build up ng fatty plaques. So class, here to summarize our 4 different lipoproteins, particularly involving your chylomicrons as we have seen in our discussion ng lipid digestion. Let's discuss. For the function again, ang boundary ng chylomicrons is for transport lang ng dietary tags from your small intestine all the way to your adipocyte, okay? So class, again, that's the boundary of your chylomicrons. For VLDL, of course, from your adipocytes, it will transport your endogenous tags from your liver adipocytes to different parts of your body or tissues. Alright, si LDL naman, it does not deliver tags, rather, it delivers your cholesterol. Also, nasan si IDL? Actually, class, similar lang kasi ang nature and property ni VLDL and IDL. Remember, si IDL ay remnant lang ni VLDL, okay? So I hope that's clear. And lastly, we have here your HDL, which reverses the cholesterol transport from your blood cells to the liver. Okay? So I hope that's clear again. For the function, si chylomicrons, transport yon ng ating dietary tags to your adipocytes. Okay? So VLDL, yung endogenous tags that are stored will be mobilized from our liver to different parts of our tissues. Okay? Si LDL naman, hindi si tags ang tinatransport niya. It is very specific to your cholesterol to different parts or to different tissues. And lastly, si HDL, the excess cholesterol from our tissues will be reversely delivered to your liver. Okay? So class, for the major lipid content, again, for chylomicrons and VLDL, both of them have very high triglycerides or tags component. For LDL, of course, because it delivers cholesterol, very obvious that it has high cholesterol or cholesterol derivatives, cholesterol esters. So similar lang yan kay HDL, but here we have your phospholipid components, okay? So for delivery kasi agad yan class for your liver. Alright, so class, in terms of the origin of our lipoproteins and chylomicrons, recall again, si chylomicrons, this is our initial lipoprotein. It acts as a transporter of your dietary tags. So class, again, ang location ng dietary tags are in your intestinal mucosal cells, okay? So VLDL from its function, it's in the liver, and remember si LDL, this is your IDL remnant or your VLDL remnant as well, okay? So finally, si HDL, ayan, ang main origins niya involve your liver and intestine. So very rich in HDL's liver and intestine. Remember class, again, si HDL ang most healthy and si major atherogenic protein naman natin si LDL. Alright, so class, in terms of their relative size, recall si chylomicrons, that's our initial lipoprotein. That's why very packed pa siya with different lipid components, your tags, your phospholipids, and your cholesterol. That's why it's also the largest. And as we go along until we reach your HDL, can you spot the trend? We reduce your sizes coming from your VLDL to IDL to LDL until we reach the smallest lipoprotein, HDL. Because again, its structure is high density na, meaning small ang size niya. Very packed na siya with your cholesterol and phospholipids. Okay, so again class, with their density and their lipid protein ratio. Ang density nila is with respect to your protein component. That's why, for chylomicrons, similar with your VLDL, they have the lowest density because of their low protein component. And on the other hand, very high po ang lipid component. So VLDL, again, low protein component as well and high lipid component as well. Si LDL, ayan, yung so-so lipoprotein natin. Moderate lipid and protein component. And lastly, of course, from the name itself, HDL, highest density, high protein component, and lower lipid component. Okay, so for their fates, ayan. So after delivery, your liver will clear your chylomicrons. Okay, so meaning it will undergo another round of transport of dietary tags. Okay, so VLDL, again, it is your IDL precursor. That's why it will be converted to IDL and all the way to your LDL. Alright, so I hope that's clear class. For LDL naman, ayan, it will be taken up by our tissues or our liver via our specific receptors, LDL. And then of course, it will be cleared up as well by our liver. And lastly, is HDL, ayan, it will be reverted back to your main sorting center, our liver, when our cholesterol are, or where our cholesterol is excreted. Okay, so again class, we will store again kasi your cholesterol or it will be excreted. Okay, so to summarize, the digestion and mobilization part ng ating catabolic pathway, remember, from the mouth, your lipid digestion will occur until it reaches your stomach for the formation of your fat droplets or your chyme, di ba? And here in our small intestine again, so yan po di ba yung main site ng lipid digestion? Yes, because this is where your bile salts and your lipases are secreted by your pancreatic cells, okay? Si bile salts again are for emulsification ng dietary tags to form your micelles. And on the other hand, lipases is for the degradation ng tags in order to repack it or to use your glycerol for energy use. Again, for our third step, that is where your fatty acids and glycerol breakdown occur, okay? So for our fourth delivery na yan class, all the way to our blood cells and liver adipocytes. Again, we need to protect the structural integrity of your lipids, particularly your fatty acids. That's why we form your chylomicron structure. So again class, very important ang chylomicron in order to prevent aggregation ng fatty acid. Because again, ayan, we need to move your lipids or fatty acids in our lymphatic system and bloodstream, okay? So here class, again, ang very specific lipoprotein lipase or your hormone-sensitive lipase will be mobilizing your fatty acids towards sa adipose tissue or your liver adipocytes, okay? So again, that's the time where your fatty acids will enter the cells for energy generation. And of course class, ang ating energy generation is a complex process. We will have another process that we will call fatty activation and transport. Okay, but before that, Ayan, we have a did you know portion. O class, if you have been body shamed, just remember, ayan, your DNA actually determines where our fat goes and comes from. So of course, it's out of our control or beyond our control yan. So remember, we have two different adipose tissue kasi si visceral and si subcutaneous adipose tissue. So si visceral fat yung exposed sa skin natin nearby. Si subcutaneous, of course, that's behind your abdominal muscle. And of course, we can classify our body type, apple shape or pear shape, depending where our fats or adipose tissue are located. Kapag maraming visceral adipose tissue, ayan, apple shape. Kapag naman subcutaneous ang localized or concentrated, that's your pear shape. Class, again, to summarize, genetics influences fat distribution in the body and mobilization patterns, like whether it comes from your visceral or subcutaneous fat storage first. Because again, class, our genes dictate the formation of our enzymes, your cytokines in particular, okay? So of course, class, again, remember this, our genes dictate where our fat goes and comes from. Clear ba, class? Again, fat loss is not just about effort always. Diba? We call it your biochemical GPS, where it will be mobilized in our body. Okay, so class, here is our next process, our activation and transport. Class, for the specific pathway or process, of course, always tandem ang activation and our transport. It is a quite tightly regulated process. So again, for our fatty acids to enter the mitochondria, depending on the length of the carbon chain of fatty acids, it can either freely enter or it can use your transport shuttles. Class, usually ang short and medium chain fatty acids can enter your mitochondria freely. So they are the less than 14 carbon chain fatty acids. Okay? Kapag long chain ka naman or you have 14 carbon chain or above, ayan, you need to use your carnitine shuttle for transport. So class, again, ang activation and transport, they are interrelated to each other. That's why it's a tandem process. So here are the processes occurring in our fatty acid activation and transport. Sorry. So first one, of course, we will activate your fatty acid. O sir, paano po? We need to have an input of energy to form fatty acyl-CoA. O di ba class, at this point, dapat you are well familiar kapag activation, we are just inputting or producing or giving your fatty acid an energy source. Okay, for membrane transport naman, specific shuttle that we use is your carnitine shuttle. Okay, hindi siya katulad ng mitochondrial shuttle that we have two types. Okay, so here si fatty acyl-CoA will be attached temporarily by carnitine and it will enter or it will aid the entry to your mitochondria. And lastly, ayan, we will regenerate your fatty acyl-CoA in your mitochondrial matrix. Sir, bakit po i-regenerate? Ano po bang na-form sa membrane transport? Class, we need to condense kasi your fatty acyl to your carnitine enzyme. Diba? Ayan, this is the specific visualization for this process. Again, here is your original fatty acid. So we need to, fatty acyl-CoA rather, so this is the activated form na. But we need to transport it all the way to your mitochondrial matrix. That's why, temporarily, in our membrane transport, we will condense your fatty acyl functional group to your carnitine to liberate your CoH coenzyme. And then afterwards, ayan, you can see that upon entry to your mitochondrial matrix, we will just regenerate your fatty acyl-CoA through the reverse process occurring in your cytosol. Okay? So here, class, I would like to emphasize, si acyl transferase 1 ang responsible sa transport from your cytosol to your intermembrane space. Si carnitine acyl transferase 2 naman ang responsible sa regeneration step. Okay? So again, they are differently localized in our mitochondrial membrane. Okay? So here, class, ang specific reactions ng activation natin. As you can see, similar enzyme lang ang nagkakatalyze ng two stepwise reaction here. Ang first one, again, we need your ATP for activation in order to form first the adenylate molecule. Let's check the arrow pushing mechanism. Here is your fatty acid molecule. And of course, your electronegative oxygen, it will induce an attack to your phosphate group, thereby forming your adenylate molecule. So class, again, we will liberate here your pyrophosphate molecule. Okay, so class, as you can see here, this is your pyrophosphate region ng ating ATP. And of course, upon formation ng fatty acid adenylate, we will use again another molecule, si CoH for activation, using the stored energy kay AMP, again, similar enzyme with your fatty acyl-CoA synthetase, and here we generate your fatty acyl-CoA. That's why your class actually equivalent to 2 ATP ang nagamit in activating our fatty acid. Or remember that info because we will need it in our ATP accounting sa fatty acid beta oxidation. Okay? Ayan, for our activation again, recall, your fatty acid will react with ATP forming fatty acyl adenylate as the main product. And then for our hydrolysis, we will use your your pyrophosphate to form your inorganic pyrophosphate that will power our second reaction and third reaction, our thioester formation. So again, here your CoA will attack your acyl adenylate, displacing the AMP to form your thioester compound. Okay? So I hope that's clear. Now let's proceed with our second step ng fatty activation and transport. So here again, class, ang main membrane transport through your carnitine acyl transferase 1 and 2 activity. Let's check. Again, this is the same visualization or diagram from our previous slide. So here again, class, important to note, si acyl transferase 1 is the one exposed sa cytosol. So it will catalyze the entry of your fatty acyl-CoA via condensation to your carnitine complex to form your acyl-carnitine, and this serves as our carnitine transporter. And of course, again, in our matrix, we have here your peripheral enzyme, si acyl transferase 2, of course, will catalyze the regeneration. So sir, ano po ang product here? Ayan, from your cytosolic fatty acyl-CoA, it will be transported through passing your outer mitochondrial membrane to your intermembrane space until it reaches your inner mitochondrial membrane, where your acyl transferase or your acyl transferase 2 will regenerate your mitochondrial fatty acyl-CoA. So again, class, ang site ng beta oxidation is your mitochondrial matrix. All right? So now for our third step ng activation and transport, again, si acyl transferase 2, it will simply transfer the acyl group back to your CoA in order to feed it to our beta oxidation. So again, class, we will generate also your free carnitine ready for another round ng membrane transport of fatty acid. So to summarize our steps or processes here under your activation and transport, ayan, class, we have here your activation again. Remember our enzyme, fatty acyl-CoA synthetase in your cytosol and your outer mitochondrial membrane. For membrane transport naman, we have two again, si acyl transferase 1 and 2, and your acyl-carnitine or carnitine transporter embedded sa inner mitochondrial membrane. A very obvious location, mitochondria. And lastly, for regeneration, again, ang responsible acyl transferase enzyme here is your acyl transferase 2 in your mitochondrial matrix. And class, always remember the regulated step, okay? Or the point of regulation here in our whole catabolic process, starting from digestion to your mobilization and activation and transport as well as oxidation, ang carnitine-mediated entry. Because once mag-enter ang ating fatty acyl CoA in your mitochondrial matrix, automatic po, okay? Naka-autopilot mode na ang ating beta oxidation. That's why here is our regulated step, the carnitine-mediated entry, okay? So class, let's proceed with our next process under your fatty acid catabolism. That is your beta oxidation. Okay. So class, again, for beta oxidation, remember, this is a sequential step or removal step ng 2-carbon of your fatty acid to form your acetyl-CoA. Class, remember ha, 2-carbon molecule po si acetyl-CoA. And here, we remove these 2 carbons via the oxidation step at your specific carbon site, si beta carbon. That's why we call it your beta oxidation. Let's locate your beta carbon here. So again, class, here is our carbonyl functional group that is also a part of your thioester functional group. Okay, let's check. Again, the nearest or the nearby carbon is our alpha carbon. And after that is our beta carbon. So that's the site of our cleavage po later in order to produce your acetyl-CoA. Class, check your manual. Your beta oxidation consists of 4 individual reactions. Again, it involves 4 individual reactions. Sir, do we need to memorize each reaction? Actually, class, no. In the board exam, their questions is more on the ATP yield. That's why after this discussion on beta oxidation, we will immediately discuss the ATP calculation. Don't worry, we summarize the important formulas and concepts that you need to know here. Okay. So first, with dehydrogenation, ayan, remember class, here in our first step, we will form your FADH2 electron carrier through your acyl-CoA dehydrogenase enzyme. Sir, ano pong nangyari dyan? Of course, we just introduced a double bond between your alpha and beta carbons to form your enoyl-CoA compound, okay? So afterwards, we will just hydrate that specific double bond to introduce naman your hydroxyl bond. That's why, through your enoyl-CoA hydratase enzyme, we were able to form your hydroxyacyl-CoA intermediate, okay? Sir, bakit important na mag-add tayo ng hydroxyl group? Because class, in order to proceed with our oxidation, dapat meron muna tayong available for oxidation or our oxidation site, which is your hydroxyl group, okay? So that's our third reaction. As you can see here, we will convert your hydroxyacyl-CoA via oxidation to form your another carbonyl functional group, and ayan, we form also your electron carrier NADH. So here class, our product or intermediate is your ketoacyl-CoA molecule, and of course, this will be subjected to your cleavage via thiolysis or the breakdown of your thioester bond. Okay, so again class, here you can see we broke down your thioester bond here via your acyl-CoA acetyl transferase enzyme. So ano pong product? Again, always product si acetyl-CoA, and here is our final product or our product that will be fed again for another cycle ng beta oxidation. So class, si myristoyl-CoA is just your fatty acyl-CoA with -2 carbons in its structure relative kay palmitoyl-CoA. So again, nag-remove lang kasi tayo ng 2 carbons in one round ng beta oxidation. So here class, again, this cycle will continue until we cleave 2 carbon units every turn where we will fully oxidize your fatty acid chain. Okay, so again, it depends to our original or initial fatty acid substrate. So to summarize our discussion here, ayan. So again, it's just a continuous cycle. So for instance, if we have a cis-16 carbon, we will produce your acetyl-CoA molecule here through different or numerous rounds ng beta oxidation. So don't worry, we will tabulate how many rounds, how many acetyl-CoA, and your electron carriers will be produced upon full oxidation ng specific fatty acids. Okay? But for now, class, let's check the summary. Again, for the function, from fatty acids, we will produce numerous or large amounts ng acetyl-CoA that will be fed to your TCA cycle when we need the energy. Okay? Class, when it occurs, again, during fasting, prolonged exercise, and low carb. So this tree, they signal that our body or blood is in low blood glucose concentration conditions. That's why, class, we need to resort to other biomolecules for our energy source because we need to maintain our blood glucose concentration. Okay? Ang tissue involvement, of course, our active organs, sa liver, muscle, and for heart, ayan, for blood pumping. And again, recall our energy products, acetyl-CoA, FADH2, and NADH. So for instance, class, for your cis-16 or 16 carbon fatty acid, saturated fatty acid in particular, we will need 7 rounds ng beta oxidation. Sir, bakit 7 lang? Because the last round, it will produce 2 molecules ng acetyl-CoA for an even numbered saturated fatty acid. Okay? So going back, of course, ayan, we will produce 8 acetyl-CoA and then 7 NADH and 7 FADH2. Class, here is the overall reaction. No need to memorize. Again, yung rounds NADH and FADH2 electron carriers, similar lang sila ng amount upon full oxidation ng fatty acid. Let's summarize. Ayan, again, for our 16 carbon fatty acid, here is our formula. So for number of rounds and your FADH2 and NADH molecule, again, they have the similar formula. So for 16 carbon fatty acid, 16 divided by 2, 8 minus 1 is 7 rounds, 7 NADH, and 7 FADH2. For acetyl-CoA class, very straightforward. Simple division, number of carbons divided by 2. Sir, ganyan ba lagi ang formula? No, class, this is very specific for your even numbered saturated fatty acid because in biological system, our naturally occurring fatty acids are saturated and even numbered. But of course, we also have kasi your monounsaturated and your PUFAS. So class, si PUFAS naman ang calculation niya almost hindi nag-appear sa board exam because it's always complicated. It's rather complicated, okay? But for monounsaturated, so this can be or this can possibly appear. But of course, very easy lang to memorize the formula for this. So class, recall, sa first round ng ating beta oxidation or the first reaction is the double bond formation. Diba class, remember? Ayan. For monounsaturated fatty acids, this reaction is actually bypassed because, well, obviously, sa structure ng monounsaturated, it already has a double bond, okay? So that's why the first reaction does not occur in your monounsaturated fatty acid. And recall, we were able to generate here your FADH2 molecule. That's why -1 po ang yield ng FADH2 for even numbered monounsaturated fatty acid, okay? So I hope that's clear. So class, again, for numbered naman na fatty acids, we will yield acetyl-CoA and propionyl-CoA. So very rare naman na mag-synthesize ang body natin ng add-numbered fatty acids. So class, recall the structure of your propionyl-CoA. That's why much easier ang ating formula for add-numbered saturated fatty acids. So class, again, less than 1 na po ang ating acetyl-CoA because again sa last cycle ng ating or last round ng ating beta oxidation, we will yield acetyl-CoA plus propionyl-CoA. Okay? So class, again, this is our formulas for add-numbered saturated fatty acid, similar to each other lang. For number of rounds, acetyl-CoA and FADH2 and NADH amounts, ayan, your number of carbons in its chain divided by 2 minus 1 will be our number of rounds and your energy products. Okay? O sir, ano pong mangyayari kay propionyl-CoA? What will happen to that specific molecule? So of course, it has different fates in your animal cell and your plant cell. In animal cell, we can convert it to your succinyl-CoA. Okay? So remember, your succinyl-CoA is a TCA cycle intermediate. That's why we can feed it to your TCA cycle still for energy production. However, for plants, ayan, we can still convert it to your acetyl-CoA plus the liberation of carbon dioxide. Okay? So again, remember, for animals, propionyl-CoA will be converted to succinyl-CoA, but for plants, they can still convert it to acetyl-CoA via liberation ng carbon dioxide molecule. Okay? So plus, ayan, we now arrive with our ATP calculation discussion. Remember, from our beta oxidation, it's not the end of our catabolic pathway, diba? We need to generate ATP kasi that's why we will feed it in your citric acid cycle. So our 8-acetyl-CoA generated from your beta oxidation will be fed in your citric acid cycle. Also, what will happen with our electron carriers? So of course, class, it will be fed in our electron transport chain or our ETC sa stage 3 ng ating ATP generation. Also, similar reaction mechanism ba yan with our cellular respiration? Yes po, ang stage 2 and stage 3, that's just similar with the TCA cycle and electron transport chain process occurring in our cellular respiration. O class, mag-account na tayo ng energy products. O check. Again, our main example here is your 16-carbon saturated fatty acid. And remember our energy products. Again, for our beta oxidation of your 16-carbon saturated Fa, we were able to generate acetyl-CoA. How many? 8 po. Alright? So of course, this acetyl-CoA will be directly fed separately to our TCA cycle. Let's compute our energy products upon feeding your acetyl-CoA sa TCA. Ayan. Remember our energy products sa TCA, di ba? 3 NADH, 1 FADH2, and 1 ATP or GTP molecule per acetyl-CoA fed in your TCA cycle. That's why here, because we generated 8 acetyl-CoA, we will multiply all of these energy products by 8. Okay, so class, straightforward, di ba? 8 times 3, 24. 8 times 1, 8. Okay, so next, ayan, we have your ATP conversion. Oh, nagkamnisya na ata kayo again. For the conversion, for NADH, that's equivalent to 2.5 ATP per NADH. Diba recall? 10 protons kasi ang napapump for your NADH electron transfer. And of course, we need 4 protons in order to generate 1 molecule of ATP. That's why 10 divided by 4, 2.5. So that's the similar rationalization kay FADH2 here. Your 6 divided by 4 will be 1.5 ATP per unit FADH2. Okay, so class, as is lang ang ATP natin here. Again, they are equivalent. 1 GTP is equal to 1 ATP. That's why, for total ATP yield, ayan, 24 times 2.5. O class, please double check ha. 60. 8 times 1.5, 12, and finally 8 times 1, 8. Done na tayo mag-account ng 8-acetyl-CoA turned into your ATP product. As you can see, very large ang ATP production coming from your beta oxidation. Lagdagan pa natin, class. So your electron carriers will be fed in your electron transport chain. Remember, 7 for FADH2 and 7 for your NADH. Similar conversion lang yan with our NADH and FADH2 conversion from the TCA cycle. That's why 7 times 1.5 is equal to 10.5 and 7 times 2.5 is equal to 17.5. Class, please double check again. And here, class, is our total ATP produced, 108. Okay, sir, yan na ba yun? Hindi pa, class. Diba recall, baka nag-amnesia ulit. In your activation step ng fatty acid, we used 2 equivalent ATP for your fatty acyl-CoA synthetase reaction, di ba? So we use ATP to form your fatty acyl-CoA. That's why, again, 2 ATP equivalent siya. We need to subtract minus 2 or 2 ATPs here in our ATP accounting. So class, ang net ATP yield natin, always remember, wag mako-confuse, 106 ATP. Because in the board exam, for the past 2 years, laging nasa item ito. Okay? So nasa biochem subtest nito. So they will ask for the net ATP yield and ang choices, increments of 2 lang. So majority ng students will answer 108 if they did not or if they failed to account your 2 ATPs used for activation. O diba, class? Very, very complicated ang ATP calculation for beta oxidation. Diba? So again, for gluconeogenesis, it's energetically expensive. So beta oxidation, it yields large amounts of energy. That's why majority, class, ng ATP that is used by our cells are coming or is coming from your beta oxidation. To summarize our net ATP yield for our even-numbered saturated fatty acids, class, please remember, again, from your C10 to C20, we have your mnemonics, diba? Crush lang muna para safe ako. Okay? So here, from our net ATP, actually, you can see may trend tayo here. So that's just increments of 14 ATP per addition ng 2 carbon chain. And with that, we can actually produce or formulate a formula here. Class, ang relevant here lang actually is for your saturated, even-numbered, and your monounsaturated fatty acids. But of course, you can also familiarize your formulas for polyunsaturated and your saturated na odd-numbered fatty acids naman. Okay? So for saturated, even-numbered, again, this is for the net ATP yield. 14 times your carbon chain divided by 2 minus 6. And then for our monounsaturated, similar lang with our parentheses. But here again, minus 7.5. O sir, ba't nag-increase ng 1.5 yung need to subtract? Again, to account for our first step ng beta oxidation, it will bypass your first reaction. Hence, minus 1.5 ATP po in terms of its net ATP yield. Class, clear? Absorb muna, mag-reflect. What are your weakest areas here? So again, class, very complex ang discussion on beta oxidation. So please take some time to revisit our discussions here. Alright, so class, to end our discussion, Here in our fatty acid metabolism, let's answer our sample problems here. So class, this is a actual or an actual board exam question. So calculate the net ATP yield from the complete beta oxidation of one molecule of your stearic acid. O class, dapat alam nyo how many carbons are there in your stearic acid carbon chain. Okay? So class, again, how many carbons are there for your stearic acid? O sir, alam ko even numbered yan eh. O, let's use the mnemonics, diba? So of course, that's a C18 saturated fatty acid. That's why, ayan, direct plug in lang yan. So ATP yield, 14 times C over 2, again, 18 carbon chain. So 18 divided by 2, that's 9. That's why 14 times 9 minus 6 is 120 ATP. So class, remember this answer. It appeared already in your board exam. Okay? So class, please double check our calculation. Use your calculator para safe and assured kayo na correct ang calculations nyo. Okay, for another example. Oh, again, here, oleic acid naman. Hala sir, parang hindi yan saturated fatty acid. Yes, actually, this is also a C18 fatty acid, but it has an unsaturation, a one double bond in your structure. So it's actually a C18 to 1, or meaning it has a one double bond in its structure. So it's a monounsaturated fatty acid. Okay, so class, use the formula for your unsaturated or monounsaturated fatty acid in particular. So here, 14 times C divided by 2, so 18 divided by 2, that's again 9. 14 times 9 minus 7.5, answer 118.5. Also, i-round up ba? Yes, because again, we do not have a half ATP in our biological system. That's why ang answer here, class, is 119 ATP. O class, clear? Okay, so good job class. Again, if you're confused, just pause the video or go back to our previous discussion. But for now, I will summarize na our discussion on fatty acid metabolism. Ang dali diba class? Very easy as well as here in our discussion on your beta oxidation. Again, ang main process here kasi is your beta oxidation. No? Ayan, for digestion again, initial cleavage of your tags to your fatty acid, free fatty acids in particular, plus your glycerol backbone occurring in your small intestine to be absorbed by your chylomicrons or by your chylomicrons, and it will be transported to our liver adipocytes mainly. Again, after your digestion, it will be stored, and kapag need na ng cells ng energy, ayan, it will be activated. For your fatty acid to be activated, you need your ATP input to form your fatty acyl-CoA here in your cytosol. And of course, this is a preparation step for your transport and oxidation. Okay, for transport naman, again class, ang mediated or our regulated step is your carnitine shuttle. Remember, we will first condense your acyl-CoA, the original acyl-CoA, to your carnitine to form acyl-carnitine complex, and then it will be regenerated to your mitochondrial or in your mitochondrial matrix. So class, again, ang locations natin, starting from the cytosol to your site ng beta oxidation, the mitochondrial matrix. So class, very important ang transport for importation ng long chain, particularly ng long chain fatty acid. O sir, si short and medium chain fatty acids, ina-activate rin ba? Yes, but of course, they do not need specific carnitine shuttles. Okay? And lastly, as we have seen very fresh from our discussion, beta oxidation, formation ng ATP eventually via your energy products, acetyl-CoA, your 2 carbon molecule plus your electron carriers, C-NADH and C-FADH2 in your mitochondrial matrix to produce large amounts ng energy, diba? So class, please remember as well here, our regulation discussion. O class, familiar sa acyl transferase 1, that is on our transport mechanism, diba? So ang activator niya, si glucagon. Meaning, kapag meron tayong low blood glucose concentration, we will resort to your fatty acids as our source ng energy. And of course, si low malonyl-CoA, ayan. Recall kasi class, si malonyl-CoA is actually our donor molecule sa biosynthesis. That's why, kapag need natin ng energy, low ang malonyl-CoA, it will activate your acyl transferase 1 enzyme. So clearly, your presence or high malonyl-CoA will inhibit naman your carnitine acyl transferase 1. Clear? For our acyl-CoA dehydrogenase, ala sir, encounter ba natin yan? Yes class, in our beta oxidation reactions. And class, that's why si fatty acyl-CoA is actually an activator of this specific enzyme. Actually, lahat ng beta oxidation enzymes will be activated by your fatty acyl-CoA. Sir, bakit po? Because again, this molecule is our initial substrate for the beta oxidation. And finally, for our inhibitor, we have your electron carriers, our energy products. So they signal to say that we have enough energy in the body. And of course, they will inhibit the process of beta oxidation and actually your acyl transferase or your carnitine mediated entry as well. Clear class? Okay, so class, absorb, reflect, and please take a break muna. Okay? So after this discussion, we will proceed now with our biosynthesis. So relatively shorter na lang ang next discussions natin here on your lipid metabolism. Okay? So kapit lang class, again, we will start with your fatty acid biosynthesis. Okay? Alright, as we have discussed earlier,